Pleasure to be here with you today and tell you a little bit about the preclinical discoveries that are happening in our program. So why work on pancreatic cancer? Well, it's, as many of you probably know, it's a disease with very poor overall prognosis. It is the fourth cause of all cancer deaths, very poor overall survival rates of only 7% of patients, less than, being alive 5 years post-diagnosis. And critically, that survival rate has only changed by 1% in the last decade. So largely, it has remained unchanged for almost 50 years now. The average survival is almost only about 6 months, and that's potentially, this is a very complex disease, so it's because of various number of reasons, but one of these is that usually it presents as advanced already at the time of diagnosis. And there are currently no curative systemic therapies for this disease. So that's really why I work on this cancer. It's because it's such a difficult to treat cancer. There is a long way to go for us, and there's lots that we really need to do. This slide just really is there to illustrate that I have a reasonable sized team, but it's not really just about my research. It's about collaborative research that we do as part of a larger pancreatic cancer program. And that's not just within the Garvan, but also includes both our Sydney collaborators, national collaborators, as well as international collaborators. And so these are some of the types of preclinical and clinical research that we have as part of a larger collaborative. And just further illustrate, so this is run both by myself and Paul Timpson within the Garvan, and then it includes our clinical collaborators, so close clinical links to largely around Sydney at the moment, Royal North Shore Hospital, Westmead Hospital, which are the two largest recruiting sites for patients with pancreatic cancer. We've also got links to personalised medicine trial activity, as well as more national collaborators. And then there's a whole range of preclinical research going on with a variety of collaborators all around the world. So why sequence? So as Carol has mentioned, so I was a part of a large cancer genome sequencing consortium. It's called the International Cancer Genome Consortium. And as part of this, we've sequenced now over 400 pancreatic cancer specimens. So why do these types of studies? Well, so we are an inquisitive bunch, so we are genuinely interested to find out what makes these tumor cells so different from the normal healthy cells within our body. But ultimately, particularly here within the Kinghorn Cancer Center, we're really interested in exploiting these differences to try to develop either better diagnostic tests to perhaps identify these patients better, or to use this information, which is largely what's done in my group, to try to design better tailored therapies for the disease. And so what we've shown now, both as part of our sequencing study as well as those of others, is that pancreatic cancer is a molecularly very varied disease. And that's just shown in this little pictorial. So it just shows you that so it's not a single disease, essentially. There are many, many different subtypes. And most genomic aberrations in this disease occur at a frequency of only 2% or less. So this is why we believe that the one drug for all patients approach, which is currently used in the clinic, it's just not the right approach to go, particularly with this cancer. We think that there needs to be a more personalized or tailored approach so that each individual patient that presents, the therapy needs to be tailored according to the biology of that individual's tumor. Okay, so I will just very briefly run over this because this is a published paper where we were able to use sequencing data directly to guide therapy. So both testing preclinically to show that particular drugs work much better, but as well as actually translating it into the clinic for a handful of patients. And so I'll just show you one of these examples. And I'd also like to run you through some sequencing data because that's always nice to learn some genomics when we're doing these presentations. So this illustrates, so this is just 40 of the initially 100 pancreatic cancer specimens that we've performed whole genome sequencing analysis of the DNA. And so what this is, and I'll run you through it, because by the end of it, you'll all be experts in looking at these types of plots. So this is a circular plot, a circus plot, and it summarizes all the genomic data for this individual tumor. Chromosomes are running, and I'll just show you, actually I'll move around. So chromosomes are running in a circular manner, and each of these lines represents a single change in the DNA code. So these lines can represent either individual letters of the DNA code that have been mutated. You could have pieces missing, so things like deletions in areas of the chromosomes. Chromosomes that are aberrantly jointed together, so these are these long lines that are going, for example, from chromosome 2 down to chromosome 16, 17, down here. So you can see all sorts of different changes happening. And one thing that became apparent to us as we've profiled a large number of these tumor specimens was that there were specific tumors that just had a huge number of these genomic changes. And what we termed them was, we essentially called them tumors with unstable genomes, or highly genomically unstable. It just means hugely rearranged, lots of different things happening. Some of the characteristics of these tumors were that they're fast growing, they're very aggressive, and they don't seem to respond to gemcitabine, which was standard of care therapy at the time. Now we've slightly shifted to a combination of gemcitabine and Abraxane, which is still not particularly effective. But what we knew from particularly breast cancer studies was that these types of tumors, because a large number of breast cancers have also been profiled, and people have observed these types of changes as well in a proportion of breast cancers. So they've found that these particular tumors may actually respond to a specific DNA damaging class of agents, things like platinum or mitomycin C. And what's also neat about these drugs is that they're old drugs, they're clinically used. Occasionally they are used in the treatment of pancreatic cancer. So we thought this would be an ideal thing to try and repurpose to treat a specific subtype. And just to show you that, so this is why these differences became very apparent to us. Not all tumors carry this signature. So this is a tumor that we've called a stable tumor genome. So you can see that there are very few lines there, so very few changes happening in the DNA of that particular tumor. So potentially, you would not want to be targeting that particular subtype with cisplatin. So there's probably something else that's better suited. So just to run you through the pipeline, so this is how the discovery kind of happened for this particular molecular subtype. So you identify the genetic defect, so that's the unstable genome. Okay, we've seen the stable genome again for comparison. You create a hypothesis. So for us, the hypothesis was that these tumors should respond to the DNA damaging agent cisplatin. We then needed to test the hypothesis in preclinical models. So that was part of my job. Within the initial, so when I came back from Holland, it was to set up these preclinical models. So we've taken, as tumor material was sequenced from the patients, we've taken parallel samples of that tumor material, implanted it into mice. So we were able to establish 80 different patient tumors growing in mice that were then available for these types of testing of novel therapies. We also, from these specimens, were able to generate patient-derived cell lines. So these are lines that are cultured in the laboratory, and they're available for large types of in vitro drug screening. So that's the first step. So we took our patient tumor-derived cell lines, ran these screens, and found, so when you plated them on a dish, add the drug on top, and then you can look at the effects on viability or how alive the cells are post-treatment. So this is just shown in this graph. So here we have an unstable tumor on the left, the unstable tumor genome, and a tumor with a stable genome on the right. We treated with two drugs, gemcitabine shown in blue, which is standard of care therapy, so just for comparison, and we've treated with cisplatin, which is the targeted agent in this case. What you can see on the left is that as our hypothesis predicted, the targeted therapy worked, and it worked quite well. So there is a lot of kill. So you can see on the y-axis, it shows you the percentage of cells that are alive at the end of the treatment. And so it shows that most of the cells are actually killed by cisplatin, specifically in that unstable tumor. So again, it highlights on the right that you really do not want to be giving cisplatin to just everyone. We're not saying that. We're saying personalized therapy for each different subtype of pancreatic cancer. And we've also done a whole bunch of other experiments to show that this is really a robust result. And so this is just shown here. So we've taken, so this is now the patient-derived xenograft model. So these are the patient tumor-derived mice models of the disease. And again, it shows you here. So it's just on the y-axis of this graph on the left. We've got the tumor volume. So we're measuring tumor volume in these mice as they're being treated with different therapies. Black is just the control, so untreated essentially. Gemcitabine shown in blue, so the tumor grows straight through, it doesn't respond, and it responded well to targeted therapy, which was cisplatin again. But the ultimate goal for us was to apply this strategy in the clinic. And so we were able to do this. So at the time we've generated this preclinical data, we had a few patients who we knew that they had that genomic signature there. They were unstable tumor genomes, and they relapsed on standard of care therapy. So that's also another problem. You can't always bring your discovery and then just test it first line. You have to wait for the patient to receive standard of care therapy, not respond to that, and then you could persuade the oncologists maybe a bit more to try something a bit more experimental. So this particular patient relapsed following gemcitabine treatment. So that's just shown here in the white. There is a recurrence. Oncologist, when we brought our results to the oncologist, was quite happy to administer cisplatin, partially also because this is not a completely novel therapy, as I've indicated before. It is clinically used, dosages are established, so it was very easy for the oncologist to feel, to feel happy about giving this drug to the patient. And surprise, surprise, actually, this patient is doing really well. So this was a complete response to the treatment. Over 3.5 years post-diagnosis, which is incredibly rare, particularly post-relapse in this disease. So that's one of the good stories, I guess. But what we'd like to do now is to really identify the sort of best treatment approach for each individual patient or subgroup of patients. And so we've been fortunate enough to acquire funding from both the NHMRC, Cancer Australia, Cancer Council New South Wales, as well as this is also done by huge support, the philanthropic support that comes to us from the Garvan Research Foundation and through our donors to work on some of the more novel therapies. And just to show you a bit of, we like to call it science fiction, so that it's not just dry pictures. This is a recent publication that we've just had using this drug called Fasudil. It's a really interesting drug because it's currently used in the treatment of cerebral vasospasms, so in the treatment of stroke in Japan. It's a vasodilatory agent. So, and it hasn't been ever, it hasn't been clinically repurposed for cancer therapy. So we thought this would be quite attractive to try and see whether this particular inhibitor, it's a small molecule inhibitor of a targeted pathway it's called ROCK. It's frequently found to be overactive in pancreatic cancer, particularly in metastatic disease. We thought it would be a valuable target to test in our models and see if we can actually improve response to standard of care chemotherapy when we use this agent. Essentially what we found was that Fasudil had, through dual effects, so this is now imaging of of tumors. So we have on one side just controlled tumor material, on the other side Fasudil treated. And it's showing in purple, we basically used advanced imaging to exploit a sort of natural occurrence where you can visualize the stromal deposition in these tumors as purple. So that's the purple autofluorescence that's detected by microscopes. And one of the key components, I guess, of pancreatic cancer is the presence of this very active, it's called stromal reaction. So these are seemingly what you might think normal cells, but they've been programmed, so they've been pulled by tumor cells to specifically to increase the ability of tumor cells to propagate essentially and form these large tumors. So if you look at a pancreatic cancer specimen, actually what you find is that there are a few tumor cells, and then there are masses of this stroma, or normal cells that have infiltrated and are surrounding the tumor itself. And when you cut through the tumor tissue or tumor specimen, it's extremely hard. It's almost like a carrot, like very, very hard tissue. What we believe that does is actually part of the role of that tissue is to prevent the drugs, so the chemotherapy, from actually accessing tumor cells effectively. So that's one potential part why pancreatic cancers may be so resistant to chemotherapy. And so using Fasudil, we were able to exploit that. So we've shown, so in the top, you can see green cells are just tumor cells that have been labeled. And purple just shows this collagen or stromal deposition in these tumors. And what you can hopefully see is that post-Fasudil treatment, you see a lot less of this purple inside these three-dimensional structures. So we've just essentially taken a 3D image of a tumor section there post-treatment. So just taking the things that we already knew from the vascular research, so the fact that Fasudil is a vasodilatory agent and it is used in the treatment of stroke, we applied it here. So we looked at on the bottom, on the bottom two images, we've looked at the vessels. So that this is now the tumor vasculature following treatment with Fasudil. And so what you can see on the left is just the control. These are very clearly defined vessels. On the right side, hopefully what you can see is that these little particles that we use to image the vessels, so they're called quantum dots, are much more are entering the tumor tissue, the surrounding tumor tissue, much more readily. So these vessels have become a lot leakier, allowing these quantum dots to essentially permeate. So what we were able to show essentially is that Fasudil makes the vasculature a lot leakier, and we use this just a very short-term priming approach treated these tumors for 3 days, gave the mice a rest, and applied chemotherapy. And what we were able to show is that through this priming approach, you could extend overall survival of these mice quite dramatically. So this is more than 50% improvement in overall survival compared to standard of care therapy. So this is just shown on that plot on the left. So we've taken a tumor that had a particularly high stromal content. So it was very purple when you analyze the tumor material. And we've treated with either, again, control. So on the left, you can see % survival. So each time that line moves down, so that's essentially a mouse that had such a high tumor burden that we, for ethical reasons, had to cull the mouse. So it essentially shows you that vehicle doesn't, obviously, saline control doesn't work at all. These tumors grow rapidly. Gemcitabine Abraxane, which is shown in the green here, I'll just point to it. It's very, so these colors are great for publication, not so great for slides. But you can see here, Gemcitabine Abraxane, so that's standard of care, very slightly improves the overall survival in these mice. And this was the priming regimen, so where we used just 3 days prior to giving chemotherapy of applying this specific small molecule inhibitor, and then applying chemotherapy 3 days after that. So I guess what we did here is a slightly different approach. So here again, we're talking about personalized treatment, but it's now based on signatures that are coming from stroma, from the surrounding tumor microenvironment. And so what the plot on the right just shows you is that we've taken the specimens that have been genomically sequenced as part of the International Cancer Genome Consortium, and then we profile them using an automated imaging approach for that collagen autofluorescence, so that essentially that purple autofluorescence on the microscope. And what we've found is that you have a range of tumors that have very high levels of this stromal deposition, medium and low. And what we're currently proposing within the Kinghorn Cancer Centre, which is, for us, it's an ideal place to be in because it's an established phase 1 clinical testing site, is we're now trying to discuss this with the oncologist to see if we can actually use this to start to identify patients that may be suitable for this type of treatment. Obviously, because Fasudil has never been combined with chemotherapy before, that's why we're looking at doing firstly the phase 1 safety testing and then looking for signals of response. So, just to summarize, I think I'm not sure how I am with time, but so in addition to some of the novel therapies that we're working on, we've also got new projects and applications that we've now put through to the NHMRC that involve a lot of, so it's either agents that are clinically used, but not used in the treatment of pancreatic cancer, novel agents that are progressing through clinical trials, but also we're looking at pharma collaborations, so things, so for example, we have a very novel DNA targeting antibody that we're trying to repurpose, again, for the treatment for specific subtypes of pancreatic cancer. And so that's sort of the kind of new stuff that's hopefully coming through and that hopefully you'll hear about in the near future. But really the ultimate goal is to apply a more personalized approach to how we treat this disease. One way standard of care therapy for everyone is just not the right way to go. And with that, I would just like to thank my direct team on the left, so that's my immediate group, as well as the APGI, the key members of the Australian Pancreatic Cancer Genome Initiative, some of the key collaborators, as well as the generous support really of the patients, the family, friends, the family members and their friends and members of the community, without whom we would not be able to do any of this research. So thanks very much, and I'm happy to answer questions.